studying for boards. Disclaimer: I do not own the rights to the images or information. This was purely made for the purpose of studying for boards. CHP5 The neuron. A typical neuron has dendrites, a cell body also known as a karyon, an axon, collateral branches, axon terminals, and synaptic bulbs. The most common arrangement of neurons lying within the neural axis is for the dendrites to lead directly into the cell body and the whole of the axon to follow the cell body. The position of the cell body can vary, however. The primary afferent neuron, also known as the first neuron in a sensory pathway from the periphery, has its cell body in the dorsal root ganglion of the spinal nerve. In this type of neuron, the cell body has slid along the axon and in functional terms is now near the end of the axon instead of the beginning as shown in Fig B. The peripheral end of a primary afferent neuron typically consists of fine branching receptor terminals, which correspond to the dendrites of an ordinary neuron. The extreme tip of a receptor terminal is specialized structurally in electrophysiological to form a receptor ending. The resetting of the neural cell body of a primary afferent neuron reflects the evolutionary advantage of removing the cell body as far as possible from the surface of the animal's body where it would be too easily damaged. No matter what the position of the cell body, the nerve impulse normally travels from either the dendritic or the receptor endings towards the axon terminals and ends at the synaptic bulbs of the axon terminals. In the next image you can see that a neuron typically has many branches. There are usually numerous dendrites and each dendrite itself may have many fine branches. Zone as dendrites around the neuron is called the dendritic field. The axon ends in several branching axon terminals. The axon may also possess one of several collateral branches, each rising from the node of RANVR. Each collateral branch also has its own axon terminals which may be numerous. On the following image you can see each terminal or branch of an axon terminal ends in a synaptic bulb. The term neurite covers the axon, collaterals, and the dendrites. The synaptic bulbs are enlargement at the effector end of the axon terminal. The surface of a typical neuronal cell body within the neural axis is encrusted with synaptic bulbs. As seen in the following figure, bulbs in this site are called axosomatic bulbs. The dendrites are similarly covered with bulbs, but the number of bulbs decreases as a dendrite divides into its fine terminal branches. The bulbs on dendrites are known as accident-Rick bulbs. Some bulbs may end on the axon terminals of the neuron rather than on the cell body of the dendrites. Bulbs in this position have been named axonal bulbs, but terminal bulbs would be more descriptive of their position. These bulbs have been regarded as an anatomical basis of presynaptic inhibition. The total number of synaptic bulbs received by a single neuron may be several thousand. Some individual neurons in the neural axis are believed to receive up to 10,000 synaptic bulbs, and a large proportion of these may have come from separate neurons. Since the total number of neurons in the human neural axis is 15 billion, the total number of possible neuronal connections is truly prodigious. The cell body of a typical neuron is very large indeed, the nucleus alone being bigger than the whole of many other individual cells, and the nucleolus is the only slightly smaller than a red blood cell. If the axon is also taken into account, the cell may be immensely long. For example, a mechanoreceptor ending in the hind foot of a thoroughbred horse leads into an axon which ascends the leg to the dorsal root ganglion and then continues along the spinal cord to end in the middle oblongate, give me a total length of about 4 meters or more. Most neurons are multipolar with dendrites and axons pointing in various directions. Pseudo-unipolar is shown in the next figure, or have only one cell process which immediately divides into two. These are primary afferent neurons with their cell body in the dorsal root ganglion. They arise in the embryo from a bipolar cell with two processes which fuse together. True bipolar neurons do a career in adult animals, being spindle-shaped with a dendrite at one end and the axon at the other end. Neurons of this type occur in the retina and the vestibulocochlear pathway. Shown in the following image, a typical neuron contains a single voluminous nucleus with a very large nucleolus. The nucleus has markedly euchromatic and therefore still standing, conspicuous clumps of basophilic material known as nissl substance are dispersed throughout the cytoplasm, including that of the beginning of the dendrites but not the axon, hillock, and axon. The nissl substance consists of aggregations of rough endoplasmic reticulum with attached and free clusters of polyribosomes. The membranes destined to form neurosecretory granules or synaptic vesicles or the cell membrane itself are synthesized in the endoplasmic reticulum and processed in a well-developed Golgi apparatus. A major proportion of the cell's metabolism is devoted to the elaboration of membranes, since essentially all the membranes contained within the dendrites, the axon terminals, have originated from the cell body or they're fully assembled are, in a precursor form, neural tubules, microfilaments, and neurofilaments run through the cytoplasm into the dendrites and axons. There are usually numerous mitochondria. Lysosomes are common, and nearly all neurons contain membrane-bound granules of brown wiring tier pigment known as lipofuscin, which are probably secondary lysosomes. The cell membrane or side of limo has the usual trilaminar structure that certain of its integral proteins and reorient themselves to create ion channels. Storage diseases arise from deficiencies of specific lysosomal enzymes and therefore result in the accumulation within lysosomes of the products of lipid, protein, and carbohydrate metabolism. Protein synthesis, the euchromatic nucleus, large nucleolus, and abundant rough endoplasmic reticulum and polyribosomes are all signs of active protein synthesis within the cell body, from which many mitochondria provide a source energy. The maintenance and repair of cytoplasmic proteins contained in the relatively enormous volume of cytoplasm and membrane proteins, bitter substances to be released at the axon terminals, also accounts for a substantial portion of this protein synthesis. The axon is also known as the axis cylinder. The term nerve fiber is sometimes used as a synonym for axon but really applies to the axon together with its sheath, for example the Schwann cell sheath in the peripheral nervous system and the oligodendral sheath in the central nervous system. This definition of nerve fiber holds good in the peripheral nervous system, where all axons, whether myelinated or unmyelinated, have a Schwann cell sheath, except sometimes for the axon terminals. However, in the central nervous system, unmyelinated fibers are totally bare and have no sheath at all, but not unreasonably are often referred to as nerve fibers. Nevertheless, the term nerve should not be used to signify a signal of myelinated or unmyelinated fiber, but applies only to a bundle of such fibers. The cytoplasm of an axon is known as the axoplasma. About 80% of the dry weight of axoplasma consists of proteins and the rest is lipids and sugars. There are thousands of different proteins, some in solution, some embedded in the membrane, and others assembled along elongated threads. Among those in solution are enzymes specific to synthesis of neurotransmitter substances. These constitute a cytoskeleton running through the cell body and penetrate the whole length of the dendrite and axon without branching. There are also membrane-bound organelles, including vesicles containing neurotransmitters, occasional small cisternae of smooth endoplasmic reticulum which storing and transportation substances such as lipids and membrane glycoproteins required in the axon terminals. Lysosomes. In mitochondria, ribosomes are absent, a fact which shows that essentially no synthesis of protein from amino acids can occur in the axon. The cell membrane of an axon is called the axonlemma. It resembles the cell membrane of the nerve cell body and possessing ion channels in its integral proteins. Like all other varieties of microtubule, the neurotubules are formed by polymerization of subunits of the protein tubulin. Microfilaments consist chiefly of a neural variant of a protein actin. Those microtubules and microfilaments are also present in most other cell types. Neurofilaments are characteristic of nerve cells and neuroglial cells and are formed from controversial protein constitutes. They are usually more numerous in the microtubules and microfilaments. Neurofilaments are about half the diameter of neurotubules, approximately 10 nanometers, Smithly 40 nanometers. Rixona transport various experiments including ligation of axons, labeling of proteins by incorporating radioactive amino acids, and direct observations in tissue culture have shown that external plasma is continuously on the move. Not only isn't moving, but it goes in both directions at the same time. Doesn't like it. Axon vesicles and other external constituents such as proteins, catecholamines, and phospholipids accumulate on both sides of the ligature. The net transport, however, it is from the cell body of the axon terminals, and this process is known as anterograde transport. Anterograde transport occurs as two types of moon, one slow about 1 meter per day and one fast about 100 to 400 milliliters per day. Slow transport is the bulk flow of the whole semi-solid axle column, which is being produced continuously in the cell body and undergoing internal dissolution of the terminals. Seems that the entire excellent cytoskeleton is neurofilaments, microfilaments, and neurofilaments move as an extended scaffolding rather than as subunits. Carried along with the scaffolding scaffolding are numerous and diverse proteins consisting mainly of many types of soluble enzymes. The function of slow transport is to renew the axal plasma continuously or to supply new exuplay by this method, which resembles extrusion of the tube from toothpaste. Fast transport, on the other hand, works by the movement of materials within the scaffolding of the axial coiling. It carries new membrane material and membrane-bound organelles. Foremost among these are vesicles, or the precursors, the best quotes, the former containing neurotransmitter substances. However, it is not necessary for all synaptic vesicles to be exported intact from the cell body, and most neuron synaptic vesicles are formed largely by local recycling memories in the axon terminal. Those the majority of neurons, the axon terminal contains enzymes for local citizens or transmitters. Nevertheless, there's still a continued need for a fresh supply of synthesized membrane and enzymes from the cell body. Much of the new membrane material exported by the cell body fails to reach the axon terminals but is deposited instead along the axon to main the axillemma. Transport from the axon terminals to the cell body is known as retrograde transport. This goes at about half the rate of fast anterograde transport. The organelles which travel by retrograde transport are again membrane-bound and consist mainly of lysosomes. Some of the contents of these lysosomes are worn out by membrane materials from the axon terminals, with your being returned to the cell body for either take-corrugation or restoration. Are there substances within lysosomes are acquired from the extracellular fluid either in small quantities by endocytosis of solutes or in much larger quantities by endocytosis substances which bind to specific receptors on the axillemma and are taken up, incorporated into lysosomes, and shift rapidly to the cell body. The latter category includes some dangerous toxins and particles, for example tetanus toxin, which is taken up and transported by virtually all kinds of neurons, and neurotrip with viruses, notably those of herpes and rabies. A lot of particles are able to multiply non-neuronal cells, but after the alleged concentrations of the virus they are effectively excluded through lack of appropriate surface receptors. On the other hand, neurons offer a sequence of mechanisms which promote the rapid arrival of virion at the cell body where it proliferates. The mechanism of axonal transport are not understood, but the participation of contractile proteins and microtubules has been considered. The contractile protein actin and myosin, the prime biological movers, certainly are present in neurons and in theory could be the source of the force needed for fast transport, microtubules merely providing a passive skeletal framework. However, the evidence for this is not substantial. Alternatively, the microtubules could provide a slow-moving track which organelles could undergo fast transport in active stepwise manner. Lumen would be provided by local sequential energy-dependent conformational changes resulting from the interaction of the organelle with a subunit on the track in a ratchet mechanism or as a sliding filament mechanism. Tubulin and actin and myosin are polar molecules. Polar moly orientation therefore transport would be possible in both directions if some of the molecular tracks had one polarity and others have the opposite polarity, explaining the simultaneous occurrence of anterograde and retrograde transport within the axon. Rapid axonal transport in both directions over distances of a meter or more through such a narrow tube as an axon is an amazing engineering achievement, with similar phenomenon intracellular motility or curve in many other cells. For example, the movement of the spindle during mitosis and amoeboid movement. Therefore, axon transport is really only an example of a highly specialized example of standard cellular activities. Anterograde transport being a step in the biogenesis of membranes, and retrograde transport being a step up the cycle of membrane turnover. The myelin sheath of a myelinated fiber in the peripheral nervous system is formed from the rolled-up cell membrane of the Schwann cells. The central nervous system, the illegal dangerous cells, form myelin by throwing out cell processes which wrap around an axon. In a peripheral nervous system, the unmyelinated axons are suspended, several together within one Schwann cell. In the central nervous system, the unmyelinated fibers are barren, totally devoid of any supporting cells. Even a myelinated fiber has regions where the axon is bare myelin. Such regions include the initial segment and the conical axon hillock. Together these form the beginning of the axon and motor neurons and interneurons. The axon hillock and initial segment have great functional importance since they constitute the trigger region of the neuron which the volleys of action potentials originate. The node of Ranvier is also bare of myelin. Each node represents the junction of two adjacent Schwann cells. The node of Ranvier also of great functional significance because they form the basis of saltatory conduction of the nerve impulse, which is characteristic of myelinated axons. The high resistance of myelin effectively seals the surface of the axon. Only at the nodes around the air and the depolarization ionic flux of the nerve impulse take place, and therefore the depolarization process has to jump from node to node, hence the term saltatory conduction, which means leaping conduction. The functional value of a leap is shown by the fact that conduction in myelinated fiber is about 3 times faster than unmyelinated fibers of the same diameter, fiber diameter, and conduction velocity. In vertebrates in general, the diameter of the axis cylinder and the thickness of the myelin sheath tends to increase by similar proportions as the thickness of the whole fiber increases. In higher mammals, the external diameter of the largest myelinated fibers is about 20 micrometers and the smallest about 1 micrometer, including the myelin sheath for both instances. The largest unmyelinated fibers have a total external diameter of approximately 2 micrometers and the smallest of about 0.1 micrometers. Since the limit of resolution of the light microscope is only about 0.25 micro, the smallest unmyelinated fibers cannot be studied with light microscope. Moreover, the distance between even the larger unmyelinated fibers is often less than 0.25 micrometers, and consequently these fibers can only be distinguished individually. The faster in conduction and nerve impulse, whether it be myelinated or unmyelinated, is a rough guide. The conduction velocity in meters per second of myelinated fibers can be estimated by multiplying the diameter by a factor of 6, plus the largest myelinated fibers of 20 micrometers have a conduction of approximately 120 meters per second or 270 miles per hour, and the smallest being approximately 6 meters per second. Is already stated, myelinated fibers conduct about 3 times faster than unmyelinated fibers of similar diameter. The conduction of large unmyelinated fibers can be estimated by multiplying by a factor of 2, thus an unmyelinated fiber of 1 micrometer diameter conducts at a speed of approximately 2 meters per second or 4.5 miles per hour. If myelinated fibers were not available to mammals, would it be possible to utilize unmyelinated fibers which would be capable of conducting at adequate velocity? Theoretically, the answer is yes, but these would have to be unmyelinated fibers of colossal diameter. A typical nerve innervating the mammalian muscle is about 1 millimeter in diameter and contains approximately 1,000 myelinated fibers from 10 to 20 micrometers in diameter. Is such a nerve, would it consist of the same number of unmyelinated fibers with the same conduction velocity? The fibers would have to be of such a massive dimensions that the nerve would resemble a thick rope of about 4 centimeters in diameter. If the human spinal cord contain only unmyelinated axons, we need to be several meters in diameter in order to maintain its current velocities. Next, we'll move on to the epineurium, perineurium, and endoneurium. The epineurium and entoneurium are typical connective tissue elements. The epineurium provides an irregular collagenous coat around the outside of the peripheral nerve, enclosing all the nerve bundles and penetrating between them. The endoneurium forms a delicate fiber collagen fibrils and fibrosites around the individual nerve fibers within a nerve bundle. It also forms thin septa which blend with the perineurium and closed groups of nerve fibers. Included within the endometrium are capillaries and other small blood vessels which are useful for estimating the diameter of nerve fibers in histologic transverse section of the nerve. The perineurium is a much more specialized connective tissue structure forming a thin but well-defined sleeve around each bundle of nerve fibers in the nerve. It consists of several concentric layers of thin epithelioid cells and collagen fibrils. The cells are attached to each other by tight junctions which form unaffected diffusion barrier to most macromolecules, thus isolating nerve fibers from noxious material in the surrounding tissue fluid. The cells also have many micropinocytotic vesicles. The perineurium is open at within like a sleeve of the jacket as shown on the following figure, thus allowing the nerve fibers access to their target tissue. The opening also allows toxins and pathogens to enter the sleeve, and this has been suspected as another route by which rabies virus reaches nerve cell bodies. Next, we will discuss the synapse. As you can see on the following figure, synapses have essentially the same components within all parts of the nervous system. The junction between efferent neurons and their effector organs, for example, between the axon terminal of a motor neuron and a skeletal muscle cell and between receptor cells such as those of the organ of Corti in the middle ear and the effects on receptor endings which reply to the cells are also essentially similar. The synapse within the neural axis forms a good model and has the following characteristics. General structure of the synaptic bulb. A synaptic bulb is an enlarged end of the axon terminal. They usually contain some mitochondria and numerous small vesicles which are believed to enclose transmitter substance. Cholinergic vesicles are spherical and about 30 to 60 nanometers in diameter and in electron micrographs appear to be empty. Noradrenergic and adrenergic vesicles are also 3260 nanometers in diameter but contain a dense core and therefore called small granular vesicles or small dense core vesicles. The presynaptic membrane. The exolemma of the synaptic bulb where it is applied to the cell membrane of the adjacent neuron is flattened and it's called the presynaptic membrane. And electron micrographs that usually appears thick and electron dense though somewhat less so than the postsynaptic membrane. Transmitter vesicles tend to be passed along the presynaptic membrane. During synaptic transmission, vesicles fuse with presynaptic membranes and open into the synaptic cleft, this being the same process as exocytosis plants. The term presynaptic membrane is also applied to the cell membrane of a receptor cell, for example in carotid body type 1 cell where it forms synaptic contact with its sensory axonal ending. The synaptic cleft, the space usually slightly widened between the presynaptic and postsynaptic membranes, is known as the synaptic cleft. As a general rule, it contains an Arizona dark material which typically lies near to the postsynaptic membrane. The postsynaptic membrane. This is the cell membrane of the adjacent neuron. In electron micrographs that usually appear thicker and denser than the presynaptic membrane. The asymmetry of the pre and postsynaptic membrane seems to be characteristic of excitatory synapses, but in inhibitory synapses the postsynaptic membrane is apparently no thicker than the presynaptic membrane. Transmitter substances. In synapses between neurons there are essentially two varieties of transmitter substances, excitatory and inhibitory, which can be called a substance and I-substance respectively. After release, transmitter substances are rapidly removed from the synaptic cleft. Acetylcholine and noradrenaline are common transmitter substances throughout the nervous system. You see the choline that is released at a synapse from cholinergic vessels is rapidly inactivated mainly by acetylcholinesterase which splits it into choline and acetic acid. The choline is reabsorbed but recycled within the Santa big bowl. Noradrenaline that is delivered into the synaptic cleft from noradrenergic vesicles is largely resorbed and recycled within the synaptic bowls. The release of transmitter substance and the onset of their effects of postsynaptic membrane both take time. This pause in the transmission of nervous activity is known as the synaptic delay. Edic synaptic delay is about 0.5 to 1 millisecond. With the advancing knowledge, a wide variety of possible neurotransmitters is beginning to emerge. Dopamine appears to be an important catecholamine within the neural axis and seems to be mainly inhibitory in action. You mean serotonin is evidently an important synaptic transmitter in some parts of the brainstem and spinal cord. Periods or their derivatives such as ADP may form another group of neurotransmitter. Several amino acids are possible transmitter substances in the mammalian neural axis. One of these is gamma-aminobutyric acid and this appears to be inhibitory. Others are believed to be excitatory. There is some evidence that in the mammalian neural axis the amino acids could be the major transmitter while the better known transmitters such as acetylcholine, noradrenaline, dopamine, and serotonin may only account for transmission at a small percentage of central synapses. Finally, a group of neuropeptides has recently attracted much attention as possible transmitter substances. These are synthesized in the nerve cell body, transported to the axon terminal, stored there and then scored vesicles approximately 18 to 120 nanometers in diameter and release on depolarization of the term endorphins and enkephalins. These are naturally occurring substances that their actions are similar to exhaustion as opiates such as morphine. They appear to be involved in neuronal pathways regulating the conscious perception of pain. All opiate neuronal receptor sites are blocked by naloxone. It has long been believed that each neuron is equipped with only one neurotransmitter. This concept would be known as Dale's hypothesis. It is now clear that this concept is not valid. For example, many pre- and postganglionic parasympathetic neurons contain neuropeptides as well as acetylcholine, and so also do many preganglionic sympathetic neurons. Likewise, many postganglionic sympathetic neurons contain neuropeptides in addition to noradrenaline and adrenaline. When more than one of these substances is released from axon terminal, it is postulated that they modulate each other's neurotransmitter activities. The release of transmitter substances is affected by the concentration of calcium and magnesium ions in the extracellular fluid. In every known secretory process, depolarization of calcium ions from the extracellular environment to intracellular channel is formed by protein molecules causing an increase in intracellular calcium. The increase in calcium ions can also be achieved by the release of stored intracellular calcium. It seems likely that in the synaptic bulb on the calcium ions promote the fusion of synaptic vesicles with the presynaptic membrane and hence include the release of transmitter substance by exocytosis. It just a disturbance in the balance of magnesium or calcium ions can produce pronounced neuromuscular disorders such as Grass staggers or hypomagnesium or milk fever, which is hypocalcemia. Curarium synthetic curare substances compete with acetylcholine for the receptor sites at the postsynaptic membrane of the motor end plate. They produce a block without causing depolarization of the postsynaptic membrane. Other drugs such as succinylcholine paralyze muscles by producing persistent depolarization of the postsynaptic membrane at the motor end plate. Botulinum toxin paralyzes by preventing the release of acetylcholine at the motor end plate. Next, we will discuss phylogenetically primitive and advanced neurons. As shown in the following figure, terminals diverge to make synaptic connections with many other widely separated neurons, thereby illustrating divergence. The dendrites of a primitive neuron, which usually have only one generation of branches, are straight rather than curved and therefore receive many synapses from relatively distant regions of the neural axis because of their widespread and numerous connections. Primitive neurons form extensive networks of interconnected neurons. In contrast, a phylogenetically advanced neuron has a long axon with only a few collaterals and axon terminals. Moreover, as axon terminal shows convergence, in other words, they tend to converge on only one or two neurons. The dendrites of this type of neuron branch repeatedly and are relatively long, but they are also curved rather than straight and therefore remain fairly close to their cell bodies. Consequently, they tend to receive synapses from regions nearby rather than far away. Neurons of this variety are characteristic of which form so-called specific pathways. Neuronal pathways which conduct only single or particular kind of information are known as specific pathways. An example is the visual system. Non-specific pathways, on the other hand, if you're a wide variety of information. An example is ascending reticular formation, in which the neuronal pathways consists of the fused networks of primitive neurons shared by impulses arising from receptors of touch, pain, pressure, temperature, and so on. And its most refined form, a specific pathway, which consists of one chains of neurons that's enabling a single stimulus to be conveyed with maximal precision to the cerebral cortex where it could be accurately identified. 121 pathways have I ever require a great wealth of neurons and her luxury with even human neural access can sell them if ever afford. For instance, even in the eye or specific pathway would seem to be especially advantageous. There are 6 to 7 million cones but only 1 million axons in the human optic nerve. Evidently, several receptor cells converge on one bipolar cell and several bipolar cells converge on one ganglion. However, it is apparently not uncommon in the neural axis of higher mammals for a specific neuron to make synapses with only 2 or 3 of the neurons. This happens, for example, in the medial meniscal sensory system of touch, pressure, and joint movement, also known as kinesthesia. Next, we will discuss external degeneration and regeneration of peripheral nerves. As you can see in the following figure, the response of a peripheral nerve fiber to transactions are shown. Aid is a normal motor nerve fiber nervating a skeletal muscle fiber via motor end plate, basal lamina, cell body, and Haskell green. In being changes during the first week after transection. In the proximal stump towards the cell body, the axon myelin sheath died back a short distance known as ascending degeneration, but the Schwann cell just above the cut survives and begins to proliferate. The neuronal cell body swells, the nucleus becomes eccentric away from the axon hillock, and the nissl substance disintegrates known as chromatic license. The axon sprouts elytra fine filaments. In the distal stump of the axon and its myelin sheath degenerate, the debris being removed removed by macrophages of the basal lamina of the original Schwann cell remain as an intact tube. The muscle cell begins to atrophy. C is 3 weeks after the cut. The proliferating Schwann cells fill the tube of the basal lamina. One external filament has wended its way to the end of the tube. Two supernumerary filaments also are within the tube. Other external filaments and Schwann cells have spread outside the tube. The cell body has returned to several months after the cut. The supernumerary filaments within the tube and the filaments outside the tube have gone. The axon has been remyelinated in the motor end plate re-established. The muscle cell has recovered. Reiterate all that in more detail. The proximal stump undergoes a sanding degeneration in which a short length of the axon degenerates and its myelin disintegrates. Cell body swells in the nucleus move to an eccentric position away from the axon helic, and this substance undergoes dissolution with a resulting decrease in basophilia of the cytoplasm, again known as comatovisis. As soon as the debris the proximal stomach has been removed by macrophages, the axon sprouts or leash or fine filaments. Schwann cells begin to proliferate and move towards the distal stump. Some escape from the cut end of the fiber. This stage could be reached within a week of the injury as shown in B. The distal stump experiences descending degeneration, also known as valerian degeneration. Silver changes begin very quickly so that by 24 hours the myelin sheath and access cylinder degenerate throughout the entire length of the fiber from the cut to its peripheral end. By the end of 2 weeks the degeneration is maximum. The myelin have disintegrated into fatty droplets and the access cylinder into small fragments. Macrophages phagocytize the debris. The basal lamina however remains intact as a continuous tube all the way to the axon terminal. Within 3 weeks regeneration is on the way. Schwann cells form the proximal stump have sealed the tube of basal lamina and several filaments from the axon only have wended their way through the tube. Other filaments have wandered outside the tube. After several months the wandering and supernumerary elements have disappeared and in the 2-1 surviving filament has been remyelinated to reach the motor end plate. Certain conditions are needed for successful regeneration. The gap between the proximal and distal stumps must be small and must not be filled with collagenous scar tissue. The two stumps must be correctly aligned. Of the various tubular structures that might guide the regeneration of axons, we tube of the basal lamina which belongs to the original fiber of the critical importance. If two stumps become misaligned so that the regeneration of axons of the proximal stomach cannot find the basal lamina tube of the distal stomach, or that the distal stump is removed as an amputation of the limb, the filaments may continue to sprout from the proximal stump, forming painful mass known as an amputation neuroma. A question of why the regenerating axons of the cut nerve enter the tubes of the basal lamina of might come up first because the tube survive the cut. The perineural sheath also survives injury and is a sharply defined tube but is obviously far too short to guide all the many regenerating axons enclosed within it to their individual targets. The endoneurium may also remain intact but is not organized into a private tubule for each nerve fiber. The Schwann cells distal to the injury are potential guidelines but they rapidly disintegrate and are phagocytized. In contrast, the basal lamina of the Schwann cells does not disintegrate nor is it attacked by macrophages. Therefore, delicate though it is, it offers every axon an accurate trail to its distant target. It has been observed that the basal lamina of muscle cells, endothelial cells, and pulmonary alveolar cells is not attracted by macrophages after degeneration of the cells, and that these cells also regenerate within their tubes of basal lamina. There is evidence that some substances found in base such as proteoglycan might be associated with promoting the outgrowth of neurites. Possibly the distal nerve segments or cell debris might have a trophic influence on regenerating axons. Because each proximal axon stump sprouts many filaments, several may enter one tube of basal lamina. Only one of these filaments is finally selected and the others disappear. Usually the entirely successful is the afferent axon grows into a tube which ends in a motor end plate, or vice versa. Function will not be restored. In the human hand some sensory deficits always persist after several nerves have been reconstructed. Next we will discuss regeneration and plasticity of the neural axis. Most neurons in the medallion relaxus survive the last band of the individual, but with advanced aging there may be slow but steady loss. The olfactory epithelium cells is a notable exception to the longevity of neurons since it lasts only about a month and it's been replaced by division of basal cells in the olfactory epithelium. The cell body of a mature neuron possesses no capacity whatever for reproducing itself, but axons in the peripheral nerves good powers of regeneration. On the other hand, axons of the neural axis have only a very limited capacity for regeneration. Thus if an axon in the brain or spinal cord is cut, the axon must dump nearest to the cell body send out sprouts which penetrate the site of the lesion, but after a week or two they stop growing. Various inconclusive explanations for this arrest has been suggested. Astrocytes may proliferate fill in the side of the lesion with a glial star. Nerve growth factors derived from blood may be unable to reach the sprouting axons or stumps, or may be blocked by anti-growth substances secreted by the neuroglial cells. One strange aspect of this problem is that axonal regeneration does occur with precision in the nerve axis sufficient in amphibian but fails in reptiles, birds and mammals. The failure of the exinus browning animals does not however mean that recovery from a lesion cannot occur. In fact, the central nervous system shows remarkable plasticity in response to injury. Functional improvement begins during the first days or weeks, but this is due to resorption of edema and blood, and the recovery of axons from sonia. Then in May and there begins a slow but progressive restoration of function even after severe traumatic injuries or vascular lesions such as with a stroke, which is scarcely ever complete but can yet be so good that remain deficits may escape even a sophisticated neurological examination. Several mechanisms appear to be involved. First, undamaged axons passing through the region of a lesion form collateral sprouts which spread out in the damaged area known as collateral sprouting. These sprouts form new synapses at synaptic sites which have been vacated previously. Undisclosed pathways in the affected region have become functional known as unmasking. Thirdly, the resumption of function may be a learning process in which the brain modifies the neuronal circuitry by altering and adjusting synaptic connections, as may be assumed to occur during any kind of learning process in the immature individual. For this to happen, patience is essential. Learning to write is a slow process in both the child and the disabled adult. For in man, motivation to recover is apparently of overwhelming importance. These mechanisms require months or years to occur, but man, the outlook is especially hopeful. And veterinary practice, the prospect of progressive but very slow recovery must be weighted against the needs and abilities of the owner and humanitarian considerations. Next, we will discuss the reflex arc basic principles. The structural unit of the nervous system is the neuron, but the functional unit is the reflex arc. Somatic reflex arcs enable to animal to react to its external environment. Autonomic reflex arcs regulate the internal environment by adjusting the functions of the circulatory organs and other viscera. The simplest reflex arc consists of only two neurons, one afferent and one affair. Since these two neurons require only a single innervating synapse, this is known as a monosynaptic reflex arc. Monosynaptic arcs are somewhat unusual in mammals, but they occur in the stretch reflex or myotactic reflex, which controls the activity of skeletal muscle. The most familiar example of such a reflex is the patellar reflex. Usually, however, several interneurons lie between the efferent and afferent neuron. A simple reflex arc begins at the receptor ending of the primary afferent axon. Receptor endings of the afferent neurons are continuous of the afferent neuron. Cell body being in the dorsal root ganglion. This location of the cell body applies to primary afferent neurons throughout the nervous system, with the only exception, namely the cell bodies of the musing cephalic trigeminal nucleus and the first neurons of the olfactory pathway. After leaving the cell body, the axon continues through the dorsal root into the neural axis, where it ends on axon terminals. These corn snake tank connections with a chain of interneurons, which in turn makes synaptic connections with the efferent neurons. The efferent motor neuron, known as the motor neuron in the somatic reflex arc, has cell bodies in the neural axis and the ventral horn in the case of the spinal cord. In a somatic reflex arc, the axon of the motor neuron passes out through the ventral root into the peripheral nerve. Its axon terminals act on the effector organ, which would be skeletal muscle fibers in this instance. The effector organ is also known as the effector. In the autonomic reflex arc, the efferent neuron is the postganglionic neuron with its cell body, and the autonomic ganglion the preganglionic neuron being really an interneuron. The autonomic effector organ may be cardiac muscle, smooth muscle, or glandular epithelium. A reflex arc can be interrupted on their pathologic conditions at any point in its pathway, as there may be a defect in the afferent part of the pathway or in the interneurons or in the efferent part of the pathway. If the efferent part of the pathway is damaged, the interruption occurs at the motor cell body and the peripheral nerve at the effector ending or in the effector organ itself, and it may be difficult to distinguish clinically between these various possible sites. Is the effector organ response of a reflex arc occurs only on the same side of the body as that which receives the stimulus, the reflex is called an ipsilateral reflex. If the effector response is on the opposite side, it is a contralateral reflex. A spinal reflex arc in which all neurons are confined to one segment of the spinal cord is known as an intersegmental reflex arc. If the reflex involves neurons of several spinal segments, it is described as an intersegmental reflex arc. One of the most remarkable characteristics of neurons within the neural axis, particularly like those with long axons, is a tendency of their fibers to cross over from one side to the other, known as decussation. This is a very primitive feature, since in all chordates from amphioxus onwards there are standing axons in the spinal cord which use a shore of white matter ventral to the central canal. In mammals, this reaching the spinal cord is known as the white shore. In amphioxus and lower vertebrates, there are also descending axons in the spinal cord which again decussate. In higher vertebrates, including mammals, many of the descending tracts decussate. It is possible that decussation arose from the coiling reflex as shown on the next image. Coiling reflex and early representatives of the chordate phylum. The coiling reflex is a defense response which enables a very simple animal to curve its body away from a noxious stimulus. The coiling reflex is the most basic of all spinal reflexes and is present in very primitive chordates. These simple relatives of the vertebrates possess spinal chains of interconnected efferent and efferent neurons, but all of these neurons are entirely ipsilateral. Efferent neurons activate segmental myotomes on one side of the body only. However, there are also interneurons in these anomals, some at the cranial end of the spinal cord and others at the caudal end, each with a long axon. The axon of such an interneuron at the cranial end of the cord decussates and sends the spinal cord. The axon of the interneuron at the caudal decussates and sends the spinal cord. Each of these long interneurons receives synapses from adjacent unilateral afferent neurons and makes a series of synaptic quaintly noxious stimulus on either side of the body, sets up the following sequence of neuronal and muscle activity. The stimulus excites the afferent neuron excites the interneuron excites the efferent neuron along the opposite side of the body, and the efferent neuron excite their myotomes. Does the myotomes contract in sequence along the side opposite to the stimulus? The result is the body coils away from the stimulus. A similar defense neuromuscular coiling reflex was probably formed by the decussation of the optic nerves in the most primitive vertebrates, causing the head to coil away from the visual stimulus. Ejaculating interneurons which mediate the coiling reflex in the most primitive vertebrates may represent the predecessors of the long ascending and descending axons which cross over in such an amazing way in the neuroaxis of the highest vertebrates. As the evolution of the nervous system progressed, increasing numbers of these long dicues-dating neurons may have developed. Ultimately, this process could have led to the evolution of dicues-dating ascending and descending tracts which characterize the neural axis of the highest vertebrates. During the course of this evolution process, the defense function of the coiling reflex must have been replaced in the higher vertebrates by the development of the local withdrawal reflex. B. Seduced reflex flexion of the ipsilateral limb in response to a noxious stimulus. Next, we will discuss the nerve impulse. Scooby seen on the following figure. Each neuron is either excitatory or inhibitory. All primary afferent neurons eat those with their cell bodies in the dorsal root ganglion, or excitatory. All motor neurons innervating striated muscles are also excitatory. On the other hand, autonomic postganglionic motor neurons supplying smooth muscle, cardiac muscle, and glandular epithelium may be excitatory or inhibitory. Interneurons within the neuron taxis are getting excitatory or inhibitory. Ion channels and gating mechanism. When a volley of impulses arise at a synaptic bulb on a resting interneuron or motor neuron, transmitter substance is released into the synaptic cleft. It is evident that the postsynaptic membrane must possess ion channels of two kinds. One allows sodium ions to pass from the intracellular fluid into the cytoplasm, that's causing an excitatory postsynaptic potential. The other type of channel is potassium channels to escape from the cell into the extracellular fluid. This gives rise to the inhibitory post by its appropriate transmitter substance. There are indications that the channel is composed at least partly of an integral protein macromolecule. The surface of this molecule possesses a biochemical receptor site which recognizes and then finds the transmitter substance. As a result of the binding of the molecule of transmitter substance, the receptor protein molecule undergoes a conformational change which opens the channel. Evidence of the ion selectivity of different channels has indicated that the sodium channel is a rectangular hole measuring approximately 0.3 to 0.5 nanometers. The potassium channel is narrower at approximately 0.3 nanometers in diameter. In aqueous solutions, water molecules become attracted to sodium and potassium ions, causing it to become hydrated. Hydrated sodium ions are slightly larger than hydrated potassium ions. The membrane potential. In all cells, an electrical potential exists whereby the inside is negative with respect to the outside. This is a membrane potential. Although it has been intensively studied in nerve cells, the membrane potential is known to be a property of plant and animal cells generally. The nerve cell is unstable and capable of abrupt reversals as an action potential. Such unstable cells can be grouped together as excitatory cells. The sodium chloride is outside the cell in the extracellular fluid, the sodium channels being positively anchored, channels being negatively charged. Potassium and largely unknown organic ions are inside the cell. The potassium ions being positive and organic ions negatively charged. Sodium chloride and potassium ions can diffuse through the cell membrane by ion channels, but the organic ions are unable to diffuse because they are too large to go through the channels. Most of the sodium ions are kept outside the cell by the sodium-potassium pump, which trades sodium or potassium ions, exchanging 3 sodiums for 2 potassiums. Most of the potassium are kept inside the C in excess of negative charge organic ions. The essential cause of membrane potential is the relative permeability of the cell membrane to potassium ions and its relative impermeability to sodium ions. Because of potassium cannons can pass through the membrane relatively easily from their high concentration within the cell, they leak rapidly from the cell, leaving the organic ions behind. The leakage of potassium ions continues until the tendency to diffuse is balanced by the electric field which is created by the residual organic ions. At the moment of equilibrium, the inside of the motor neuron is approximately 70 mV negative with respect to the outside. Its difference of 70 mV is the membrane potential. Essentially has been created by the potassium battery. The excitatory postsynaptic potential. Characteristics of the excitatory postsynaptic potential. The postsynaptic membrane lacks the ability to undergo the rapid alterations of an activation and regeneration which produce the trains of action potentials typifying impulse activity. Instead, the postsynaptic membrane a response to generator potential. This is a depolarization which in comparison with that of the action potential is relatively small, relatively long-lasting, decremental and degraded. He reduces the membrane potential from its resting value of -70 mV to the threshold of the action potential closer to 60 mV in this content. Decremental means of progressively decreasing intensity of depolarization both with time and with distance form the synapse. Graded means that the depolarization can be varied by amplitude by interaction with other excitatory postsynaptic potentials, thus allowing summation, and inhibitory postsynaptic potentials disallowing partial or complete cancellation of excitation. Despite its decremental character, this graded depolarization given adequate summation may spread far enough over the surface of the nerve cell body to activate the specialized trigger region. This converts the sustained decremental depolarization into the explosive non-decremental action potential that characterize impulse activity. The trigger region lies in the axon hillock and initial segment of the axon. Does the release of excitatory substance and the resulting excitatory postsynaptic potential resembles a stone thrown in a pond, a series of concentric ripples spread across water. The ripples are sustained that are decremental with time and distance. When excitatory substance is released into the synaptic cleft, ion channels are open which allow the ingress of sodium ions through the postsynaptic membrane. This reduces the resting membrane potential below 70 millivolts negative. It seems likely that the release of excitatory substance causes a nonspecific increase in permeability to small cations, thus permitting these ions to flow along their concentration gradients. However, the concentration gradients are such that predominant flow will be the ingress of sodium channels. Next, we will discuss summation. If a single excitatory impulse arrives at anchor on, the resultant excitatory postsynaptic potential will probably be too small to produce an action potential. The single excitatory impulse is then called a subliminal impulse, but the arrival of a rapid succession of subliminal excitatory impulses may lower the postsynaptic potential significantly to trigger an action potential. This is because the excitatory accumulation or summits. Spatial summation happens when the subliminal stimuli arrive at the neuron simultaneously but along different axon terminals. Temporal summation occurs when the subliminal stimuli arrive in close succession along a single axon terminal. These characteristics can be illustrated by throwing a stone into a pond. The ripples caused by a single stone may be too small to reach a floating trigger. Stones when flung simultaneously into a different part of the pond may together cause enough disturbance to rock the trigger mechanism into action. This would be spatial summation. Finally, a series of identical stones thrown into the water at one place but in very rapid succession may again activate the trigger. This would be temporal summation. Facilitation. The arrival of a continuous series of subliminal excitatory stimuli may be insufficient to reach the threshold of the trigger region of the axon but may nevertheless place a cell body in a condition of facilitation. In this condition, the postsynaptic potential could have been driven down from -70 and maintained at a closer level to -16 volts in relation to the exterior of the cell. The arrival of one or more volley of excitatory impulses could now by further summation of excitatory postsynaptic potentials lower the postsynaptic potential just a little more so that the threshold of the trigger region is reached. On the other hand, if the neuron had not been already facilitated, the additional volley of excitatory stimuli alone would have been insufficient to undo an action potential. In the analogy of the pane, the surface is already in a condition of disturbance. One more stone, although in and of itself only very small effect, made and rock the trigger to its threshold. Next, we will discuss the inhibitory postsynaptic potential. Characteristics of the inhibitory postsynaptic potential. The release of inhibitory substance into the synaptic cleft causes in the temperature of postsynaptic potential. This increases the membrane potential upwards towards -80 millivolts. The postsynaptic membrane is now hyperpolarized. Inhibitory postsynaptic potential resembles the excitatory postsynaptic potential in being small, relatively long-lasting, decremental, and graded. So long as the inhibitory postsynaptic potential is maintained by the arrival of veil of inhibitory impulses, the neuron is held at a state of inefficient. During this phase of hyperpolarization, strong excitatory stimuli may be required to reduce the postsynaptic potential to 60 negative threshold of an action potential. Ionic basis of the inhibitory postsynaptic potential. The inhibitory release into the synaptic cleft increases the permeability of the postsynaptic membrane, specifically to the smaller cations such as potassium. Thus, a further egress of potassium ions occurs to the postsynaptic membranes. On the other hand, slightly larger cations such as hydrated sodium, the diameter of which is greater than that hydrated potassium ions, are unable to pass through. There is evidence that between neurotransmitters such as gabapentin act by increasing the permeability of the cell membrane to chloride ions, which are normally at a lower concentration inside the neuron than in the extracellular fluid, and then workflow of chloride ions hyperpolarizes by postnet begin admission as just described, but also by presynaptic inhibition. Presynaptic inhibition may depend on the existence of inhibitories oxonosynaptic bulbs, or more precisely inhibitory terminal synaptic bulbs. Such inhibitor bulbs would not on didn't write of an excitatory neuron but on one of the synaptic bulbs of the excitatory neuron. The inhibitory fiber could then depolarize the excitatory synaptic bulb, thereby blocking its ability to release excitatory neurotransmitter substance. The inhibitory fiber would therefore attack by limiting the amount of transmitter substance released by the excitatory synaptic bulb. More recently, however, this anatomical basis of presynaptic inhibition has been questioned. Other more complex explanations have been offered. Role of inhibitory neurons in the neural axis. There are very large numbers of inhibitory neurons in the neural axis. Some of them, such as Renshaw cells, which automatically inhibit a motor neuron in the ventral horn, act individually. Others are grouped together to inhibit some particular activity, an example being the neurons of the pneumothoracic center, which inhibit the inspiratory drive of neurons in the medullary respiratory center in order to contribute to the rhythm of breathing. Yet other inhibitory neurons, including many in the cerebral cortex cell, have widespread and diffuse effects on the neural regulation of locomotion and posture by means of reciprocal innervation. The principle of this mechanism is that when the motor neurons innervating a muscle reflexively stimulated by a volume on a synaptic efferent impulses from a muscle stretch receptor, the motor neurons innervating the antagonistic muscles are simultaneously inhibited by the same afferent veil. Does a monosynaptic afferent bodily not only initiates muscle contraction but at the same time relaxes the opposite musculature. Disinhibition. In some regions of the neural axis, inhibitory neuron makes synapses with a second inhibitory neuron. When these synapses release inhibitory substances, i.e. inhibitory neuron is prevented from inhibiting its own target neurons. This allows this target near us to become dominated by excitatory stimuli. An indirect effect of this type has been called disinhibition. The result of disinhibition is excitation. An example of disinhibition in the neural axis is the inhibitory action of the lateral medullary reticular formation on the medial medullary reticular formation, which is itself inhibitory. The net result is excitation of the ventral horn motor neurons associated with the medial medullary reticular formation. Next, we will discuss the receptor potential. The receptor ending has certain structural and functional properties which, not surprisingly, are essential similar to those of the dendrite of the interneuromotor neuron. In response to a suitable stimulus, the receptor ending develops a receptor potential. This resembles the excitatory postsynaptic potential of a dendrite in being a relatively small, relatively long-lasting, decremental, and a graded depolarization. Looking at the following figure as an example. Moreover, like the ginger, is a receptor endings of an afferent axon is unmyelinated, although sometimes it retains a finished one-cell covering. Furthermore, the receptor ending requires a triggering region to convert its initial decremental depolarization into impulse activity. In the interneuron and motor neuron, this region is the axon hillock, an initial segment of the axon. The analogous region on a receptor ending has not been formally identified, but it's probably the first node around here, and the axon is myelinated, or the segment of the axon immediately central to the actual receptive region motor neuron. The receptor potential is always excitatory, never inhibitory, whereas the postsynaptic potential dendrites can be either. The end plate potential. The effector ending of the axon which innervates skeletal muscle makes a synaptic contact with its skeletal muscle fiber. The region of the synaptic contact is named the motor end plate. This can be seen in the following figure. In a motor end plate cell membrane of the axon, effector ending is the presynaptic membrane. The sarcolemma of the muscle fiber is the postsynaptic membrane. Transmission from the axonal ending of the muscle fiber is achieved by the release of neurotransmitter substances such as acetylcholine. Just as in neuroneuronal synapse, the postsynaptic membrane of a muscle fiber responds to the excitatory influence of the transmitter substance by undergoing a relatively small and long-lasting but decremental depolarization, which in this instance is known as the end plate potential. Furthermore, trigger region is present on the sarcolemma of the muscle cell, presumably somewhere near the motor end plate, which converts the end plate potential into an action potential resulting in contraction of fibers. However, the motor end plate differs from a neuroneuronal synapse in that the end plate potential is always excitatory and never inhibitory. In this respect, the end plate potential resembles the receptor potential of an afferent receptor ending. Next, we will discuss the summary of the decremental potentials. There is much in common both structurally and functionally between the dendrite of the interneuron and motor neuron and the receptor ending of the primary efferent neuron. Structurally, these components are really all dendrites, and therefore it is to be expected that they should share functional properties. Also belonging to this functional group are the cell membrane of skeletal muscle fiber in the region of the motor end plate. The cell membrane of the cardiac muscle fiber adjacent to its autonomic efferent axonal endings is also completed along with the cell membrane of smooth muscle cells in the region of its autonomic efferent axonal endings. Relatively small, long-lasting decremental potentials characterize all 5 of the cell membranes. Furthermore, all 5 of the structural and entities require a specialized trigger region to convert the decremental potential into explosive motor neuron. The trigger region is the axon hillock and initial segment. In the case of the myelinated afferring axonal ending, it is probably the first node around the RR. In these instances, the trigger region therefore resides in an unmyelinated part of the axon. The trigger region of muscle cells has not been identified. Please send our similarities, structural and functional, between these 5 varieties of excitable cells. There are, however, some notable differences. The postsynaptic potential of interneurons and motor neurons and the end plate potential in muscle cells are all induced by a chemical transducer action, as in the receptor potential of a chemoreceptor afferent axon. On the other hand, the receptor potential of a mechanical receptor is induced by mechanical distortion of the cell membrane or axolemma. Other types of receptor endings such as thermoreceptors transduce yet other kinds of energy. Most important differences reside in the fact that the decremental potential may be excitatory only or either excitatory or inhibitory. Thus in the 5 structural entities which have been listed, either excitatory or inhibitory potential occurs at the postsynaptic membrane of the interneuron or motor neuron and at the cell membrane of the cardiac muscle and smooth muscle cells. In contrast, purely excitatory potentials occur at the receptor ending of the primary afferent neuron and the cell membrane of the skeletal muscle in the region of the motor end plate. Next we will discuss the action potential threshold. The cell body and dendrites of the interneuron and motor neuron are likely to be continually subjected to excitatory substances and inhibitory substances released by excitatory and inhibitory synapses respectively. If inhibitory stimuli predominate, the neuron becomes hyperpolarized and is dense inhibited. Is excitatory stimuli prevail, the postsynaptic potential may fall from a resting value of -70 to about -60 millivolts. At this point, the threshold of the trigger reaction is reached and an action potential results. The role of the trigger region. The action potential is initiated by the trigger region, the pacemaker region or generator region, of a neuron or muscle cell, and interneurons and motor neurons. The trigger region lies in the axon hillock and initial segment of the axon. Its position in axonal receptor endings and muscle cells is not clearly established, but in the former it is probably in the first node around the air when the air afferent axon is myelinated. In the trigger region, the relatively sustained but decremental excitatory postsynaptic potential is converted into the intermediate non-decremental action potentials which characterize impulse activity. Non-decremental means that the impulse produced in this way are conducted all the way to the axon terminals without any decrease in the degree of depolarization. The ionic flux of the action potential, as shown on the following figure, at the instant when the threshold of the action potential has been reached, the axolemma becomes highly permeable, specifically to sodium ions which rush through. This makes the inside of the axon positive with respect to the outside of approximately $50. That's the potential within the axon has abruptly altered from the -70 to approximately 50 millivolts, a total change of about 120 millivolts. This potential change is termed action potential. Once begun, the action potential continues independently of the stimulus. That's a change in the intensity of the stimulus has no effect on the amplitude of the action potential. Furthermore, an axon reacts to any stimulus either by an action potential or by remaining silent. Hence it follows the all or none law. Nevertheless, action potential of an axon does not always have the same amplitude. Various factors can change the energy reserves of an axon and thus alter the amplitude of its action potential. However, for any particular set of external conditions, the action will always respond maximally to any threshold stimulus. The action potential passes along a non-myelinated axon like a series of small explosions along a train of gunpowder. Each small explosion in gunpowder, thus the ionic flux occurring at a particular point on in the axolemma, disturb the adjacent region of the axolemma, increasing its permeability to sodium ions. And in Russia, sodium ions occurs in the newly distributors region. This in turn disturbs the next part of the axolemma. Thus the action potential moves along the axon in a self-propagating manner. In myelinated axons, the action potential jumps from node around the air to node around air. The sleeping induction is called saltatory conduction. When the action potential ends, axolemma C move out. The membrane potential therefore returns rapidly to its resting membrane potential. Thus the axon has lost some potassium ions that is made of fast recovery to its original electrical state and is ready for another impulse. The sodium potassium ions are re-exchanged later during a slower recovery. I mean to the sodium potassium pump. There are enough ions in one axon for a vast number of impulses. It is estimated that only 1/1,000,000th of a potassium ions in an axon escape during a single action potential. Refractory periods. Why the axon is fully depolarized or during the action potential cannot be very excited. This phase of depolarization is known as the absolute refractory and lasts approximately 0.5 milliseconds. During the recovery phase which follows an action potential, there is a brief overswing of the axon where it becomes temporarily hyperpolarized. This is a relative refractory. During this phase, another action potential can be induced, though less than normal amplitude, but this is possible only if the stimulus is considerably greater than the resulting threshold value. Excitability is progressively restored during the relative refractory period. The refractory characteristics of axons impose a limit on the number of impulses which an axon can transmit per second. If its absolute refractory period is 0.5 milliseconds, an axon can be driven continuously at rates not exceeding 1,000 impulses per second. Concerning water closets, summation, threshold, all or none characteristics, and refractoriness are illustrated by the flushing of a water closet. Transducer mechanisms of receptors. The receptor ending is the site of the transducer mechanism which converts the energy of the stimulus into the energy of the nerve impulse. And mechanical receptor converts kinetic energy into the nerve impulse, whereas a thermoreceptor converts heat energy and a chemoreceptor converts chemical energy into a nerve impulse. Essentially, a neuron which is responding to a neurotransmitter substance is acting as a chemoreceptor. Some receptor pathways begin with a receptor cell. Examples include the neuroepithelial cells of the spiral organ of Cardi, the vestibular apparatus, and the retina. These cells act as a transducer and release transmitter substance at the synapse with their afferent back-on. In the case of mechanical receptor endings, the ending is distorted by the mechanical stimulus. This is shown in the following figure. Distortion stretches some part of the cell membrane is causing the receptor potential. Possibly B stretching reduces the lateral attractive forces between the constituent molecules in this particular part of the cell membrane, reduces the resistance to the flow of ions across the membrane. Alternatively, the stretching may dilate pre-existing ion channels. The transducer process appears to be extremely focal. Several different spots along the receptor membrane may be activated simultaneously with spatial summation. The amplitude of the receptor potential is proportional to the total area of the membrane which is activated. Simple mechanical receptor endings consist of bare receptor endings, sometimes with a minimal covering of Schwann cell cytoplasm. So receptor endings are distorted in direct proportion to the force of the mechanical stimulus which is applied to them. So long as they are distorted, they develop a receptor potential. The amplitude of the receptor potential determines the frequency of the impulse produced by the trigger region, as the frequency of the impulse discharge is directly proportional to the degree of distortion. This is known as chemical receptors, as in the baroreceptor reflex from the carotid sinus or the Herring-Brewer respiratory stretch reflex. Complex laminated mechanical receptors consists of a central receptor ending enclosed by concentric lamina which are slightly separated by the extracellular fluid, as shown in the following image. A laminar thin cell processes belonging to a pair of Schwann cells. The lamina interdigitate much as the fingers of two hands can be interdigitating. Outside the lamina there is a fluid-filled space. This is in turn surrounded by a ring-like series of cylinder capsular cells. The Pacinian corpuscle is an example. The whole structure, including the external name in the lamina enclosing it, has an inherent elasticity which endows it with a tendency always to assume its resting, essentially circular shape. If the entire receptor complex is suddenly compressed externally, a pressure wave is instantly transmitted through the fluids within, distorts the receptor ending. This distortion generates a receptor potential which in turn releases a train of action potential. As in a simple mechanical receptor ending, the intensity of the receptor potential and hence the frequency of the resulting action potentials is determined by the area of the receptor ending which is distorted. If the external compression is sustained unchanged, the inherent elasticity of the receptor ending and its adjacent lamina causes these structures rapidly to resume their original shape. Thus they achieve this by displacing the fluid of the fluid-filled space even though the external capsule is still compressed into an oval shape. The receptor ending itself is now no longer distorted and therefore the membrane potential quickly returns to its resting value and the receptor axons fall silent. It is therefore known as a fast-adapting receptors. If the external compression is then suddenly released, the whole receptor complex brings back to its original circular shape. The returning movement of the external capsule and outermost lamp sets up another pressure wave in the fluids within, which distorts the receptor ending of the second time. This generates II receptor potential, which releases another train of action potentials. However, the ending and its laminate quickly resume their resting shape, so the ending again reverts to its resting potential and the receptor axon once more falls silent. This type of complex laminated mechanical receptor therefore fires on the on and the off of compression but is silent in between. Search receptors are capable of following rapid vibrations. They respond to essentially the rate of pressure change rather than the magnitude of the pressure. Neuroglia. The next image gives examples of neuroglia. In addition to the thousands of millions of neurons which make up the central nervous system, there are about 5 times as many supporting cells in your outglia of cells. There are 3 types of neuroglia cells named astrocytes, oligodendrocytes, and microglia. The first are divided from their neuroectoderm, but the microglia are mesoderm in origin. The neuroglial cells for the supporting framework of the central nervous system. They probably also play a part of the transport of gas, water, and electrolytes from the blood plasma to the nerve cells and the removal of the tablets. The majority of intracranial tumors arise from neuroglia from different varieties of gliomas. Astrocytes form a continuous single-celled layer immediately beneath the pia mater and covers approximately 85% of the basal lamina of the blood vessels within the substance of the central nervous system by means of their vascular feet, infused with the ependymal cells. This they form almost a complete mosaic over the deep blood vessels. Because of the way in which their feet enclose the capillaries of the central nervous system, they may well contribute to the blood-brain barrier. Their normal function is probably to monitor and by taking part in the transport of substances to control the contents of the intracellular space. If neurons died within the narrow axis, the astrocytes multiply and tend to form a glial scar which may obstruct regenerating axons. Oligodendrocytes form and maintain myelin in the central nervous system, resembling Schwann cells of the peripheral nervous system. However, the central nervous system each illegal dendrocyte encloses several axons, whereas the Schwann cells in the peripheral nervous system wraps only one myelinated axon. Microglia. As their name implies, these are small cells. They function as macrophages and rise from monocytes of the bone marrow. They migrate into the neural axis from the bone marrow.